Okay, we're back in the microbiology laboratory now. Since there's two of us here, we're talking through masks, so hopefully you can hear us. What we're going to show here is how to streak a plate to get isolated colonies. Okay, this is a really important technique in the microbiology lab when we have a sample that may have lots of different microbes in it, and we need to be able to separate them out and ensure that we're just working with one pure culture. Okay. So we're going to do this with an inoculating loop. Victoria Walton here, our fabulous biology lab tech, is going to demonstrate this for you. But before she does, I just want to show you how it's going to work. Okay, so imagine this piece of chalk is our loop, and we've got a sample of a lot of bacteria on there. Probably in the real world, it might be millions of different bacteria and a little bit of volume on our loop here. So we're going to start near the edge of our plate, right? And we're going to go back and forth. And as we're doing this, we're dropping off bacteria all along the way. We're going to go maybe a quarter or a third of the way across the plate. And then we're actually going to flame our loop, and we're going to kill everything that's left on there. Okay? So the concentration of bacteria here has gone from very high at the start to a lot lower here. So we take our sterile loop now, and we start near where we ended, and we drag it in there a couple times, and then we stay away, and we are spreading the bacteria out further. So now there's even fewer bacteria down here. And then we rotate the plate, and we do the same thing again, right? So our goal is by the time we get out here, we're dropping individual bacteria onto the surface of the plate. And those are going to grow up, maybe if we use our green chalk here, those are going to grow up into individual colonies. All those colonies, all the bacteria in that colony, which might be millions of bacteria, have all come from one starting cell that we put here on the plate. So we have tons and tons of bacteria here. We'll show you a plate in just a second. Then a lot fewer bacteria here and individual colonies here. I'm going to show you real quick the plate that Victoria is going to start with. It's a plate that's a special kind of agar that allows us to see E. coli as purple colonies. So you see it here, trying to see how I can show you well. Here the bacteria are very dense. Here they're less dense. And then over here, they've grown up as individual colonies. So Victoria is going to start with bacteria from an individual colony, but millions and millions of them, and she's going to streak it out to give new colonies on the plate, each one of which came from a single bacteria. So she flamed her loop. Right now, she actually just stuck it in a blank area on the plate to cool it down. And now she's going to take a sample from a colony, and she's going to spread it on that plate. So you see she's starting by the edge, going back and forth. And then she's going to flame her loop. So that's killing everything that's left on there. And she gives it just a little bit of time to cool down, stabs it in the agar again to cool it down. And then she's going to streak again. So now she's going from here, streaking them out again, and then rotating the plate and streaking it again. Now she's holding it up so you can see it. That's not how she would probably do it in the lab because it's kind of awkward. But basically what she did is she spread all the bacteria out, and we're going to stick this. Sorry, I'm going to sneeze. Going to stick it in the incubator overnight at 37 degrees. And that's going to let the bacteria grow up in the colonies. Okay? Thank you, Victoria.
You're welcome.